This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode number 370, recorded on December 31st, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
Good morning.
It is morning. It's 30 degrees Fahrenheit, -1 Celsius here, cloudy, humidity 78%, and not much else going
It's
on.
cold there.
Yeah.
Here it is 8 Celsius and cloudy, and we've had rain all week here, basically. This is the first cold day, but it's not really that cold. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
I bet you have similar weather to
It is. It's 39 Fahrenheit, and it's so strange. There's this really, really bright light coming out of the sky, and I don't quite know what it is. It's kind of yellowish, bright.
Oh, you mean the sun?
Oh, is that what that's called?
You really have sun?
Yeah.
Oh, we are clouded over here.
Yeah. It's been foggy and cloudy for I don't know how long, and then all of a sudden the sun broke out today. It was just wow.
You didn't have any snow, right?
Oh, we got snow. It's hard to tell how much of it is snow and how much is sleet and freezing rain. We got the nasty wintry mix that I had to scrape off the driveway.
Yeah, we didn't get anything. We got a little bit of ice one night this week, but it was gone by 10 a.m.
Oh yeah, my Wovle broke.
Oh no!
What did you do?
Well, the past 2 winters, it's been horribly, horribly abused because we've had these wretched winters, and I've been shoveling feet and feet of snow with it. So it has been very, very heavily used. And I went out to scrape the schmutz off the driveway the other day, and I started pushing it, and it got one load, and then I pushed it into the second load, and it went crack, and the blade on it is cracked.
Is it, excuse me, is it a plastic blade?
It's a plastic, the shovel part of it is plastic.
I see.
So that just wore out.
Getting a new one?
I haven't decided if I'm going to replace the blade on that or if I may finally give in and get a snowblower.
If you can, do you think you can just replace the blade? Will they sell that as a part?
I'm pretty sure I think they sell most of the parts to it individually.
Well, remember, you save the environment, you save your back. There's 4 things you save. You save money.
Right.
Anyway.
Right. You don't save your back over a snowblower, though.
You don't think so?
Yeah. Last winter it really started to sink in because all my neighbors have snowblowers, and they'd get out there and we got I don't know how many feet of snow. And the trouble with the Wovle is after you get your first 3 or so feet of snow, the banks of snow that you've been throwing it on are too high to throw a Wovle load onto it.
Right.
And so then you're back to shoveling with the regular Backsaver shovel that I also picked.
I just got 2 titles out of our little chat. I got the Wovle broke and the trouble with the Wovle.
The trouble with Wovles.
The trouble with Wovles. That's even better.
So we had that rain, freezing rain, sleet thing, but I used the alternative shovel, the yellow one that you recommended,
yes.
Alan. So I have 4 shovels. So including the Wovle, depending on what the conditions are, I choose one or more of those.
Right. So, yeah, I ended up finishing. I have 2 of the yellow ones, one with the extra handle attachment. Ended up finishing with that. Then I also have the Wovle, and when things get really nasty, I have a square tip plain metal shovel that I can actually chip ice off the driveway with, which comes up.
Kathy, you have a driveway?
Yes.
Do you have to shovel the sidewalk in front of your house also?
I do, but remember I live in this neighborhood where we use the public radio funding model, and we have funded for the entire neighborhood to have our sidewalks cleared by a brush machine.
Right.
Yeah.
Nice.
Yeah.
Well, that's good. Well, in fact, folks, we are going to talk about viruses on this show. We will not spend the entire...
This is not a snow clearing infomercial.
This is our year-end episode. Today is the last day of 2015.
Yes.
Wow. Did that go quickly? No, it went the same rate as every other year, right?
But it seemed to go quickly.
Did it seem for you to go quickly? Yeah.
Yeah.
Some people say quickly, some people say slowly.
Well, as you get older, your perception of time compresses, I think, because each year is a smaller proportion of your total life experience.
Well, I don't really have any impression on how it went. I did a lot of traveling.
When you're 5 years old, a year is 20% of the time you've experienced in your entire life, and it seems like a really, really long time.
Yeah.
And then when you're 50, it's 2% of your total life experience, and it seems like a shorter time. That's the explanation I heard, anyway.
Well, we have a year-end episode, which means we're going to talk about cool things that happened in the year before. But first, a few other things. First, I need to tell you that this episode of TWiV is brought to you by ASM Microbe 2016. If you're thinking of going to this meeting, which is taking place in Boston from June 16th through the 20th in 2016, you will need to get your abstract in by January 12th. That's the deadline. This will be the combined meeting of the ASM General Meeting and ICAC. They used to be held separately, and it will be in Boston. It's going to be a great forum for cross-disciplinary study in the presentation halls and in the integrated exhibit and poster hall. It's designed to spark collaboration and discussion. They're going to have people doing basic microbial sciences and clinical microbial sciences at this meeting. If your work touches microbial sciences, you should go to this meeting. But more importantly, we're going to do a TWiV and a TWIM at this meeting. So you should go and come by, and even if you can't sit through the session, you should say hello. We're going to do... So the Friday night we've got the schedule. The Friday night of the meeting, and there is only one Friday night, we'll be doing TWIM at 2:45. And then Saturday morning, Alan, this is unfortunate, 8:15 a.m.
Ooh, boy.
I'm sorry. I understand if you can't make it.
I'll make the effort. I'll see what I can do.
At least you won't have traffic.
It's Saturday morning?
Yes.
Okay. I won't have to take a kid to school at least. Well, it's summertime too.
June 16th.
Actually, school may still be in session.
Yeah, it could be still in session. It is
depending on
early
how many snow days we get.
But
yes, I should be able to pry myself out of bed at whatever hour in the morning that has to be.
Okay, good. So anyway, those are two good reasons, but there are hundreds and hundreds of other reasons to go to ASM Microbe.
Yeah.
And if you're going and you want to register, go to asm.org/microbe2016. That last part is one word, microbe2016. And by the way, if you're interested in sponsoring an episode of TWiV or any of our other podcasts, we do This Week in Microbiology, This Week in Parasitism, This Week in Evolution, Urban Agriculture. What am I forgetting? Is that it?
Sounds like all of them.
Microbiology, parasitism.
Yeah, they're all at microbe.tv. And we have quite a large audience of listeners who are really interested in the life sciences, a focused audience, many, many thousands of people. You should send an email to sponsor@microbe.tv, and we can talk about sponsoring you. Yeah. It's very affordable.
You sponsoring us.
You sponsoring us, yes.
What did I say?
We don't sponsor anybody. You said we can talk about sponsoring you.
Oh,
no, we're not offering money.
Yeah, you can sponsor us. We don't have any money to offer.
That's right. That's why we're asking for sponsors.
Yes, the sponsorship money, when we get it, I shouldn't really talk about this, but we've done sponsors. I haven't sent invoices yet for many of them, so I haven't gotten paid. But when it does, it goes towards our monthly expenses for the podcast hosting and so forth.
Right.
All right. We do have a bunch of follow-up. Even though it's Thursday, and everyone knows we record on Fridays, we got a lot of follow-up. The first is from Mohammed with the subject line, Camels Win Santa's Heart. Dear Dr. Racaniello and TWiV team, this is Mohammed, your biggest fan from Saudi Arabia, Riyadh in particular. I've been listening to TWiV since 2011. It all started when I searched the word science in the iOS podcast app The rest is viral history. TWiV keeps me constantly informed about recent advances in virology and vaccinology. I've been thinking of writing to you and your fabulous team for some time now. The latest episode 369 was the straw that broke the camel's back. Allow me to answer a few of the questions raised during the episode. As to how camel meat tastes, it is very similar to beef, but not as fatty, yet still tender. The preferred meat cut is the back, sirloin, and chuck, and he sent a picture, so we know where that is in a camel. The rest is preferred ground for other dishes. We mostly cook it by boiling, roasting. Pressure cookers are frequently used too. As for camel riding, dromedary camels are ridden behind the hump. It's currently 10°C in Riyadh with 40% chance of rain. As a side note, last week and many times in the past years as well, it snowed. I know. Please see attached pictures for Mr. Camel. You sent a picture of a camel in the snow.
This camel in the snow picture should be the episode photo.
Yeah, sure.
Really good.
Last week I put a camel with a Santa hat.
Yeah.
I thought that was cute. I also have breaking news to you guys. Santa is upgrading his drive from reindeer to camel. Evidence in the picture attached. I guess global warming contributed to his decision. Happy holidays to you all. Wishing you a great even year, Mohammed. You see, we know he's listening because we talked about even years. Anyway, we'll put all the pictures up, but thank you, Mohammed. That's very nice. The straw that broke the camel's back. And this camel, I mean, I presume that's in Saudi Arabia, right?
Yeah.
The one with the snow on it?
It looks rocky.
No.
Is it rocky there?
It's I guess they have mountainous regions.
Yeah,
and if you have high enough mountains, you can get snow at any latitude.
It looks like an inch or two.
Yeah.
And he's covered with it. Yeah. Wow, cool. Thank you, Mohammed. All right, Alan, can you take the next one, please?
Sure. Steen writes at 1:37:46 in the last episode. Thank you for doing the timestamp there. Vincent asks, does PLOS allow web scraping? Do they have an API? PLOS articles are under a CC BY license and therefore can be freely downloaded and reused. And he links to that. Presumably, they do not want people to scrape and rescrape too much to avoid hammering their servers. And he provides a link about terms of use for PLOS. Then says PLOS has multiple APIs and links to them. There are interfaces to these APIs from popular programming languages and provides another link for those. Michael Eisen, mentioned earlier in the show, has said in various places that frustration with restrictions on mining data from full-text articles drove him and Pat Brown to co-found PLOS. So there you go.
Presumably the terms of service are not as onerous as those from Elsevier, right?
Well, they're definitely not. I mean, because they allow reuse, this has been much discussed, that you can repackage content from PLOS in various ways and use it for just about anything you want as long as you give them credit.
So I'm looking at the terms of use, and he provides a link for that. I'm just wondering, all of it's written now, and then all of a sudden you get to a part where it's all in capital letters, like they're yelling at you.
Right.
Why do they do that?
I guess they want to emphasize that.
And then it goes back to regular, and then it goes back to capital. It's ridiculous. Online, for years, people have always felt that when you type in capital letters, you're yelling at people. Shouting, right?
Yeah.
All right, thank you, Steen. Kathy, takes the next two.
Trudy writes, where's the image of the Lego bacteriophage? So she couldn't find it, evidently. It worked for me. It was your pick of the week on 369, and it was the second image in the contest winners. So
right.
I'm not sure.
It's there, Trudy.
Right. The link is an article in New Scientist inspiring winners of New Scientist's Lego competition. First article image is a heart, and then the next image down in the article embedded in the text is the phage.
Okay, and Lily M. writes, on the topic of runny noses, I recently read a comment saying, nose cream keeps nasal passages moist when you're traveling, so you don't get tiny fissures in the membranes, preventing viruses from getting in. This doesn't intuitively sound right, but I'm not of a science background, so I thought here would be a good place to ask for clarification. Can anyone smarter offer their thoughts? Okay, so I think, getting tiny fissures in the membranes is probably the red herring in all of this. And I asked an ID doc, and he thought it sounded bogus. And then I Googled a little bit and found that GSK had something called Bactroban nasal, or mupirocin. Pyrocin, okay. And that's mentioned as an antibiotic cream to be used for MRSA. So what's the M in MRSA? Multi-drug resistant.
Multi-drug, yeah.
Staph aureus. Okay, and so so there's a skin ointment, and there's even this Bactroban nasal that you can put in. And then maybe Lily M heard something related to another site that mentions the fact that when you fly, the humidity is pretty low in the airplane, less than 15%. And so they advise to stay well hydrated and drink plenty of water. Steam from hot drinks can be good, so you could order coffee or tea or hot water. You could use a saline nasal spray or even petroleum jelly. And just to remind you that I'm not that kind of doctor, so this is all information off the internet. Do what you want, but probably staying well hydrated on an airplane is a good idea.
Yes.
I don't think viruses need a crack in the mucous membrane to infect. They will bind the mucosal surface cells, right? So cracks in that. Maybe there are other reasons not to have cracks in your nose.
Well,
as you say.
You don't want a nosebleed.
Right. I misinterpreted membranes. I was thinking cellular membranes, but you're right. If you're just talking mucous membranes, then yeah. Okay.
Right. And I think the Bactroban is for fairly specialized uses. This is not something for you to just stuff in your nose before you go on the subway. That's a specialized product for like hospitalized immunocompromised patients, I think.
Right.
So illuminate me. A nose cream, you would actually put this in your nostrils, right?
Right.
I hope this is done in the privacy of your office or home.
And they suggest using cotton swabs.
I see.
Right. It's probably just repackaged Vaseline.
Yeah. So why is the air so dry on a plane, Alan? Because
as you go up in altitude, the temperature goes down. Mm-hmm. And then in order to make it survivable inside the airplane, you have the heat on. So you're taking cold air that has a low absolute humidity and heating it, and you get a very low relative humidity in the... So how much of the air in the cabin is actually taken in from the exterior during the course of the flight? Is any? It depends on the airplane. I see. So there's a continuous turnover of the air through the ventilation system. I believe the newer aircraft, in order to conserve fuel, they recirculate a fair amount of the air.
Mm-hmm. So they don't have to heat it, right?
So they don't have to, because every time all the air you take in, you have to heat, and then, you're just sending it back out. So, and in addition, you add drag when you have any kind of an air intake. But I think it's something like 25% turnover over, an hour or something. So I saw that figure somewhere, but that's certain commercial airliners. The older the airplane you're flying, the more fresh air you're going to get.
Interesting.
There's a Cessna 152 that I rent at my flight school that I actually had to stuff one of my gloves into a vent hole that kept opening because I had this cold air blasting in my face last time I flew.
High tech.
Yeah.
So, a lot of home heating systems or furnaces, you can add a humidifier.
Yeah.
And would that be so hard to do on a plane, or would we then risk everybody getting Legionella? Or
anytime you talk about adding something to an airplane, you have to go through the whole process of approving the addition of that device to that particular airplane. Mm-hmm. And it's very much like getting a change approved for manufacturing a drug. Mm. So you think about the approval cost for that and all the tests that have to be done, and this is one of the major reasons airplanes are so expensive, is that everything has to be approved by the aviation authorities, and they are very, very particular. And then you have design problems too, like if you decide, okay, we're going to add a humidifier, it's going to weigh 10 pounds. All right, well, now we're going to have to modify this because this structure is not strong enough, and that's going to add another 30 pounds, and then we're going to have to modify this, and there's this whole cascade, and the bigger the airplane, the more complicated that is.
Right.
I must say, when I land and they open the vents, I love that breath of fresh air. It's always
Yes.
great. Oh, you can tell,
Yes.
especially in the summertime, right?
Right.
Nice. All right. I think that's part of why you feel lousy after a long flight, right? It's low humidity and
Low humidity, little bit lower oxygen. I think most commercial airliners, they only pressurize the cabin to about 6,000 to 8,000 feet.
Mm-hmm.
So you're sitting at an effective high altitude.
Yeah, right.
And you're sitting. You're locked into your seat, and
I've tried walking, but there's not a lot of room.
not a lot of room.
And you go. There's
no
always
place.
somebody in the aisle, and
Yep.
And in fact, it's hard to walk.
Yes.
You bump into the seats. You bump into people who are hanging their arms out. Boy. Yep. All right. Next one is from Wink, our friend from Atlanta, who writes, TWiV team, another great podcast. Aren't they all? Thank you, Wink.
Thank you.
We love doing every one of them. As Vincent pointed out, the particular stresses that trigger herpes simplex virus to reactivate probably evolved to promote transmission, completely ignoring the anthropomorphism admonition. I wonder if cut nerves, fever, and slamming of doors suggest to HSV, let me get out of here before my host is dead. And UV light makes HSV think my host must be awake now and more likely to contact another uninfected host. Yes, slamming of doors. I want to know what the sensor is for that. Well, it's your ear, of course, but how does it get transmitted? It could be, right? Mm-hmm. Who knows?
Yeah.
But putting it anthropomorphic, is effective, but it does bring up some issues, which we've talked about. And I'm glad you know that, Wink, so obviously you have listened. Wink is author of something called No Germs Allowed. Does anyone know anything about that? Let's look it up right now. Real time, No Germs Allowed by Wink Weinberg. No, I don't get it on Amazon.
No germs allowed. How to avoid
There it is.
How to avoid infectious diseases at home and on the road.
There it is. Oh, by Winkler
Weinberg, MD.
In the revised and expanded edition of this classic guide. I didn't know you were a classic, Wink.
Wow.
What year is this written in? The original one. Publication 1996 originally. This is a 2004 edition.
848 pages. This is a substantial book.
And it's a deal. Kindle deal at $18.95. Well, we'll put a link to that. Oh, link. We'll put a link to it. Sorry, I get excited when I can make funnies, because I don't usually do that. All right, next brief one is from Dylan White on Twitter. The pregnant camel joke came from the Zootopia preview before the Star Wars movie. At least that's where I heard it. Yes, thank you very much. That's where I heard it as well. What do you call a camel with 3 humps? Pregnant. Okay, Alan, can you take the next one, please?
Oh, now you got me.
All right, if you want to wait, we can. It's fine. We can have a...
I'll be done in a second.
Kathy, you can take the next one, please.
I could do. Esper writes,
Right.
Hi, TWiVomics. I hope this email finds you all well and free of pathogenic viruses. My name is Esper and Callis, an ID specialist and professor at the Division of Clinical Immunology and Allergy, University of São Paulo, Brazil. I've been addicted to TWiV since a friend from the University of Wisconsin participated in the GBVC episode, David O'Connor, episode 260. Since then, I've never missed one episode. After long silent listening, I decided to write for the first time, motivated by the ongoing events in my country potentially related to the Zika virus. In the last episode, Emma wrote about events taking place in the small town of Itapitim, state of Pernambuco, northeastern Brazil, where I will describe a bit later in this email. Before, let me bring some background information on the current situation. Most believed Zika was a largely benign virus causing a self-limited disease, clearly described in episode 368. Its circulation was documented after an outbreak was noticed in the state of Bahia, northeastern Brazil, by a group led by Guilherme Ribeiro, a talented young infectious disease scientist from Fiocruz, and he gives the link for that free access paper. However, things started to get awkward around October 2016 when a single hospital in Recife, northeastern Brazil, and some other practicing obstetricians and pediatricians from the region started reporting a mounting number of microcephaly cases in newborns, later confirmed by the National Registry of Newborns. The numbers are astonishing. The graph below depicts the number of cases per year prior to the surge in 2015. Only this year, 2,975 cases were reported by December 26th, the vast majority in the second semester of the year. Cases are concentrated in the Northeast with 2,608 cases, including 40 stillbirths or short-living newborns. And in response to the situation, the Brazilian Ministry of Health has declared a national public health emergency, and he gives a link for that. The Brazilian Ministry of Health has been presenting updates every week. He gives a link to that. It is important to observe some imperfections in these numbers. One, there may be an over-reporting after the news made the big media, suggesting an association between microcephaly and Zika virus. Two, the criterion to consider a microcephaly case has been changed after the current epidemic from 33 to 32 centimeters. This is because 33 centimeters of head circumference is sitting in the 10th percentile of newborns at 40 weeks of pregnancy, and the adjustment would bring the limit to the 3rd percentile, increasing the specificity to detect a true microcephaly case. This may result in an over-reporting at the beginning of the epidemic. The association between Zika virus infection and microcephaly was suspected since the beginning, when Brazilian health authorities ruled out other potential causes, together with the fact that the microcephaly epidemic followed Zika virus spread. Further evidence was the 2 positive RT-PCR for Zika RNAs in 2 amniotic fluids obtained from 2 pregnancies of microcephalic fetuses, and a stillborn microcephaly case with positive tissues for Zika RNA. In fact, French Polynesia went back to their records and also noticed an increase of microcephaly case reporting following their epidemic by the same virus strain in 2013 and 2014. Now, Zika virus transmission has been detected in several countries in the Americas, and he gives a link to that. Although strong epidemiological data suggest the association between Zika virus and the microcephaly epidemic, a causal link between the virus and the disease is still lacking and is limited to a few case reports. Many questions still remain. Does the virus damage embryonic neural tissue? What is the percentage of fetuses getting infected when the mother acquires Zika virus during pregnancy? Does the stage of pregnancy interfere with virus ability to be transmitted to the fetus and the development of neurological effects? Are there other neurological defects related to Zika virus infection? Is there another cofactor involved, such as malnutrition or other concurrent infection? All these questions are exceedingly important to provide counseling to pregnant women and those who are planning to become pregnant, especially in northeastern Brazil. In fact, Brazilian authorities have been recommending avoiding pregnancy until this situation is further clarified. The microcephaly epidemic impact is unimaginable. It is a tragedy. These children are compromised for life, and the impact on their families is beyond any prediction. Back to the story sent by Emma. A small town in the north of the Pernambuco state named Epidemic has almost 14,000 inhabitants and has recorded 11 cases of microcephaly in the past 3 months. This very same town has been suffering from a prolonged drought since September 2013 when the last reservoir went dry. Perhaps the storage of clean water or the limited resources has led to the best environment for arbovirus spread and the development of microcephaly. But the Zika virus's impact may be reaching further. An increase in Guillain-Barré syndrome cases has also been noticed in the northeast of Brazil, possibly related to the epidemic. Several groups have been trying to establish animal models to study the interaction of Zika virus with neural tissue. The forthcoming developments are critical to better understand the virus immunopathology and confirm or refute the association between the virus infection and neurologic damage in fetuses and in the infected host developing Guillain-Barré syndrome. Many things still shrouded in mystery. Keep up the good work. I will keep on listening. Esper. P.S. number 1. New York Times published today an extensive article on the microcephaly epidemic in Brazil, and I read that. It's a front page story. P.S. number 2. If I may, here's a suggestion for Weekly Picks. An article from the most recent New Yorker introduced me to the Foldscope project. It is based on the making of a microscope that's origami-based, developed by a research team at the Prakash Lab at Stanford University. It's a fascinating gateway to make the microscopic world much more popular. And I think we've talked about the Foldscope before, and I've put in the link also to the Foldscope materials paper that's open access in PLOS One. So back to the Zika story. I didn't write down the statistics as of today, but they're a little bit higher even than what Esper reported. And what really struck me is that the incidence of the microcephaly is 18 times higher than in 2014.
Yes.
So it's just remarkable.
Yes. I mean, anytime there's some supposed epidemic like this, one of the things you worry about is increased reporting, but the increase is just really, really extreme and really, really sudden in this case, and it seems like there's something really going on here.
Mm-hmm.
Yeah, I mean, clearly Zika is implicated, but as everyone says, we'll see as we do more epidemiology and
maybe we'll be
stuff.
doing Zika sit reps in the coming year.
I do think that Zika will be a big story next year.
Yes.
We have an email later about Zika. People, and I think one of the points made in this email is important. People are trying to establish animal models to study the interaction with not only neural tissue but with fetuses, and that would be important to illuminate what could be going on in people. A colleague of mine came in earlier this week because she has a daughter who is pregnant and had spent a good amount of time in Brazil in this area, and she's very worried. She didn't get sick, and the fetus is normal in head diameter, but there can be other, and this is another thing I meant to point out, besides microcephaly, who knows, even if in babies who are born and physically look normal, there could be other issues with the virus if it's in fact replicating in the infant, right?
Mm-hmm.
Well, it's probably a bad idea to get an arthropod-borne infection while you're pregnant.
Yeah.
Mm-hmm.
If you can avoid it.
Yeah, but in many places of the world, very hard to avoid. Many places people get bitten thousands of times a day, right?
Sure.
Okay, on a lighter note, the second contest, we had a winner in our first textbook contest, and I will have, it reminds me I could ship that today. No, I didn't bring it in. Oh, well. But we had a second contest, which is now over officially. Why I listened to TWiV. 280 characters or less emailed in. And some of these are quite nice. I think we should go through them quickly. The first one here from Alexander, his answer is, well, first of all, I need to know what the weather is in South Carolina, Florida, Michigan, and San Diego. That's it mostly, which you could do otherwise, but other reasons. I appreciate the discussion and analysis by people with experience dissecting papers and figures. I'm interested in the findings that don't impress you as the ones that do. The arched eyebrow of skepticism that only comes with expertise. Lots of things sound good. Only people with experience are able to say sounds good but doesn't work. And I'm particularly interested in what you don't know. I found the paper you did a while back on horizontal gene transfer in an Antarctic lake fascinating. That was TWiM 68, because every one of the panel said they didn't understand the mathematics of the analysis. Hint, get a mathematician. Even Elio said he didn't know, and I thought he knew everything. I enjoy the contrast between TWiV, TWiM, and TWiP. TWiV is the most inside baseball because of Racaniello's expertise. TWiV tends to assume a bit more knowledge, and typically Vincent knows the people and work being reported on. With TWiP and TWiM, Vincent doesn't always know the details of the critters being discussed or the investigators, so he's more the voice of an educated curious. That tends to make those two podcasts more accessible. Nomination for Outstanding Episode, TWiV 161, Concerto in B, guest Gabriel Victora. Just outstanding. This episode manages to elucidate 4 distinct subjects, none simple. 1, maturation and selection process of B cells. 2, photon excitation microscopy. 3, complex math required to infer the fates of B cells from intermittent observations. 4, the recapitulation of natural selection in the lymph nodes. Awesome. Gabriel was a great guest, and all the panel were in top form, clarifying and amplifying subtle points. Goes on your greatest hits album. Kind regards, and thank you so much for all you do. Such fun. We should make a greatest hits collection at some point. Get people's
was way past 280 characters.
Oh yes, I think just the first
the first line
was the 280.
Yeah, we may want to summarize some of these because
sure. But I thought some of these points were great.
Yeah, some of these are great.
And it is true. We do Insider Baseball on TWiV. I try not to do that as much as I can, but it's hard because it is our field,
Right.
But Alan straightens us out, right?
Yes, I try.
All right, Alan, take the next one, please.
Okay, Matthew writes, patient in, patient out. Headache, send to CT. Seizure, give Keppra. Fever, culture, X-ray, and maybe a TB test. No, refer to ID. Autoimmune disease must be genetic. Steroids. This is what we know, but I think it will take a lot more if medicine is to progress. Thanks for your podcast. Matthew sounds like an ER doc.
Does. Kathy.
Buck writes, I'm trying to make CRISPR ants. TWiV helped me see the possibilities of molecular biology. Part of the reason I switched from ecology to genetics. Ants are fascinating. Gives a link to an XKCD comic, but their viruses are almost unknown. Principles of Virology could help me to our new genetic tools, use our new genetic tools to study viruses in our ants.
Mm.
Well, if you want crisper ants, you need to dry them first, and then you dip them in a little flour before you put them in the fryer.
Yeah. Ooh.
Here's one from Elizabeth. I like TWiV on the treadmill and when riding the T, yet I listen while doing TC. I've made many cell lines and spent hours in the hood, but I've got TWiV, so as Rich says, all good. Please send edition 4 and then I'll have success, remembering to say cells produce, not express.
And I think we have a winner here.
That is the winner. We all like
Yes.
that one.
Yes.
And it really is good because it gets on. Well, TC, by the way, is tissue culture.
Yes.
And speaking of the Principles of Virology edition 4, when you guys were looking up Wink's book, since I had just found it for my class, I looked up the box that describes why we can't anthropomorphize viruses. And it's box 1.8, page 18 in chapter 1. Many scientists have succumbed to the temptation of ascribing various actions and motives while discussing viruses. While remarkably effective in enlivening a lecture or an article, these characterizations are inaccurate and also quite misleading. Okay, so viruses can't employ, ensure, synthesize, exhibit, display, destroy, deploy, defend, reprogram, avoid, retain, evade, exploit, generate, etc. Viruses can't, etc.? Okay.
Oh my.
Surprised an etc. made it in there. Yeah, the problem is that you end up thinking like a human, and that's not what viruses do.
Yeah.
They have their own goals.
But there is...
Yeah, right. That's even bad. That's
There you
even
go.
bad.
But there's a... I think
There are other reasons that viruses undergo certain chemical reactions.
as somebody who explains science for a living, I have to kind of step in a little bit and defend the
You're going to lose, man.
uses. I know I'm going to lose.
I'm just kidding.
But what I would say in defense of this is that while viruses do not have intentions and motives and such that would cause them to do verbs, there is a logic to what they do. And that logic can often be expressed most succinctly in ways that we would express behaviors.
I'm not sure there is a logic.
There is a logic.
To a human view, there is, but
there's an evolutionary logic that drives the virus to adapt in particular ways.
I don't think so. I think we think there is a logic. We
You think
sometimes don't know what the selective forces are. You know, I mean,
But we know that there are selective forces.
Yes,
and we
of
know
course.
that they have to have driven the virus to maximize its reproductive potential. That's the fundamental law.
okay, that's fine.
And so there is a logic behind what the viruses are doing, and I think there is value in perhaps not in scientific papers, or textbooks, but in explanations like TWiV, or in writing for a more general audience, or even a general interest article for a specialized audience. There's some value in talking in terms of motivation. It helps get the idea across.
I often see it in the popular press, and
Of course.
often the context is just not right. It's saying things. First of all, I don't see that it enhances the explanation for the most part, and often it's wrong because we don't know I agree with you. There are selective forces, but often people get the selective forces wrong because we anthropomorphize things. You know, viruses want to be more virulent. You know, how do you know? You just don't know that.
No, they don't want to be more virulent. They want to reproduce.
Exactly.
And that is actually, okay, they don't have wants, but there is a logic driving them to reproduce, and that could be expressed as a want, for want of a better way of putting it.
Well, it certainly, as that quote that Kathy just read says, it's much easier to anthropomorphize and makes it more interesting for people.
Yeah, and I think
it's harder to come up with an alternative. I agree.
Well, and so I wanted to ask specifically, so viruses don't synthesize. Okay, so if a DNA virus doesn't synthesize virus DNA, then do you end up using passive voice for?
Well, that's the problem, and people don't like having a passive. Exactly the problem. It becomes passive, and therefore people
cumbersome.
Do it. Yes, yeah, agreed. But we want people to know what
I think it's good. I think it's good for a textbook to lay that out and say, look, there are downsides to anthropomorphizing, and this is not, in a formal sense, the way we should be putting things. Like if you read any philosophical literature, they'll go into great detail about defining terms and use very seemingly obtuse language to state simple concepts, but it's because they want to precisely capture what they're expressing.
Yeah.
And I think that's appropriate in a textbook, and to highlight that when you do anthropomorphize, you should have a good reason, and you should be very careful that you don't start taking it too far.
In fact, I think in an earlier edition of the book, it may be still there, we said go through an interesting exercise would be to go through the text and find all the anthropomorphizations we didn't catch. Right.
You still have it. It's still there in box 1.8 in the 4th edition.
Yeah, because I'm sure there are some.
Yeah.
All right. We have a bunch more here. So here we have a person who works in a fly lab and listens to TWiV, My Biology 101. And this one was liked by 2 people. So, Alan, why don't you read Joy Rell's?
Joy Rell says, my reason for listening, I became infected by the TWiV virus, and the only known treatment is to listen to the podcasts. I started listening to after my Biology 101 instructor recommended it. I was struggling to memorize terms and found that listening to people using them helped. Thanks for helping me pass.
Yeah, that's a good point, right? Conversation is a good way to learn.
Right.
Now, the next one is from Elena, who actually won the first contest.
Yes, and is therefore disqualified. But
she did write a limerick.
Yes.
Mm-hmm.
And she works for Do-It-Yourself Bio in Toronto, a hackerspace. Wow.
Oh.
Mm-hmm.
Education and Outreach Coordinator. Kathy, you want to read Elena's?
Sure. Bringing science to the masses, each week's like virology classes. Though you'll chase away some with Alan Dove's puns, for the rest of us, discomfort passes.
Very good.
And I have to say, it's a little scary to start off a limerick with words that rhyme like masses and classes.
Yeah. Yes. I like this one from Nicola, who wrote, X brought me here, but why did I stay? Where horses more common than zebras, where mice are all liars, and monkeys tell tall tales, while dogs, cats, and people get fevers. There's Alan and Dixon, Rich, Kathy, and Vincent, more questions than answers, plaque assays, cell cultures, friends all to those plagued with bad sickness. Well.
Yes. A lot of poetry.
Yeah. Yeah.
This guy listens while waiting for his proteins to fail to crystallize.
Yes, that's good.
Brian likes to listen to TWiV to update his Stacy Schultz Cherry name drop counter.
Do we drop her name a lot?
It comes up. You ask her, you send her questions and stuff.
And then Joseph says, I research correlations between meteorological conditions and excellent virology discussions. Well, okay, not. But
actually, this next one, Alan and I liked from Kurt. I could toil for years and never describe why I appreciate TWiV better than Dixon neatly summed up biology, and perhaps in a stroke of serendipity, everything else. Life is a fractal. The closer you look, the more you see.
Yes.
Did he say that? That's pretty good.
I think yes.
It's pretty profound. Yeah.
Thereby proving the monkeys and typewriters thesis. Sorry, I should be nicer to him.
Right. Right. The next one is from Daniel, and it earlier got an honorable mention from me. He's just discovered TWiV about a month ago, and he's listening to TWiV, TWIP, and TWIM, and seems to have turned the corner and felt like he now wants to do microbiology, even though he's been an undergraduate biochemistry major.
Cool.
So that was good. And then this next one, both Alan and I like.
Yes.
Alan, why don't you read it?
Stephen, must stop zombie apocalypse. Need the virology books to develop a vaccine. Discovered zombies don't like cherry trees. Currently surrounded by zombies. Delivery by skid loader not possible. Have Dr. Dove airdrop books. Weather cloudy with 100% chance of zombies. Always a good time.
That's really good, actually.
See, it's quite good.
Yeah.
Yeah,
Very telegraphic, but
yeah, a lot of memes in there.
yeah.
Yeah, that's well done. I like that. Let's see. Kathy, you liked carols.
Yeah. Let's see. I listened to TWiV in order to hear Vincent pronounce Italian names and in eternal hope of hearing him say Reduvid bug. There just isn't enough TWiP to get enough of it. Another reason to listen is that Alan's punning is almost as good as Vincent's pronunciation. So, Vincent, how do you say that bug?
Reduvido.
Ah, okay. I've not ever heard that before.
I never said it on TWiV. Well, there you go. I usually say it on TWiP. Yep.
Yeah.
It's a wonderful name, right?
Yeah.
Not a great bug for us, but...
Right.
You want to read parts of Paul's, Kathy?
Sure. Paul wrote a long thing because he turned off his recording before he got to the part where it had to be 280 characters.
Yeah, right.
So he goes, it's basically telling about how he's become interested in AIDS and HIV, the death of Freddie Mercury when he was 11 years old saddened and confused him. And then he earned a master's degree in veterinary virology, studying the PRRS arteriovirus of pigs. And then he went on a solo backpacking trip in Tanzania and Uganda, saw and learned more about HIV and AIDS. And then he sequenced genomes of clinical flu isolates during the H1 and N1 epidemic season and subsequent seasons. He's now a PhD student at University of Toronto and studying HIV. So he's kind of come full circle there. And he wanted to point out that it was World AIDS Day last Monday, but I always thought World AIDS Day was on December 1st, and that was a Tuesday. So I'm not sure about that. It probably wasn't as much in the headlines because they were dominated by David Price signing a $270 million contract with the Red Sox to throw a ball. Just imagine what $270 million could do for basic research, let alone for providing medicines to people who don't even have money to buy food. And so then he wrote back and said, "Oh, I'm so sorry. I was running an essay and I paused the broadcast right at the very end before you said to keep it to 280 characters or less." So he summed up the World AIDS Day thing in less than 280 characters. So
it's been with us since episode 1.
Oh, that was what really grabbed my attention. I forgot to mention that. Thanks. Yes.
And I have to say, there is a little bit of a hypocrisy here in the TWiV team insisting that people keep things brief, but we had to do it that way.
Well, as you see, many people just ignored
it. They just ignore it.
And that's fine. That's fine. I mean, even the Twitter contest, we picked one where they sent in a JPEG.
Yes, a JPEG in order to bypass.
Because we break all our rules. And so what the hell? Anything else we should bring up?
Marion needs the book set to help teach underserved students. Because I am poor, is the way she put it. A good reason. David says, undead genetic material from life's primal soup stood at the cradle of mammals and through target practice keeps humans healthy.
TWiV provides a daily dive. Well, not daily. Yes. So all these letters and all the ones we read, by the way, every week I put letters on a web page, and it's usually microbe.tv/TWiV/TWiV-370-letters. That's the format. And you can just substitute the number and find them, or you can go to the link in the episode. I don't know if anyone ever looks at them, but they're there for the record. I think many people write wonderful letters, and we should put them up. Mm-hmm. Now, this is the end of 2015. In 2015, we did TWiV's 318 to 370. That's 52 episodes, one a week. And we did 12 road shows. We had 4 in-studio episodes with guests and 13 episodes with video, which is cool, at least once a month. So we've been busy. And I want to thank a bunch of people for making all this possible, because I know by the time we get to the end, if I thank you there, you won't be listening anymore or you'll shut it off. I want to thank Jennifer for doing the timestamps every week for us. We appreciate that. She usually emails them to me Sunday evening.
It's awesome.
And it's a lot of work. You have to listen
and
stop and write down. Even if she's working, she has to interrupt to doing that. And she has done it every time since she started.
And it was really great for getting things for this episode where we're going to talk about our favorite episodes of the year. And for me, I could just go to the timestamp place in the recording and listen to that. So that was great.
This was a case where a listener said you should put timestamps in. We read that, and she volunteered. So thank you, Jennifer.
Great.
And I realize you will not do it forever, because when you get your PhD, you'll probably stop, but maybe we can get someone else. I also want to thank the American Society for Microbiology for support of TWiV. They help us with our bandwidth costs. And also, when we go to meetings that they run, they support us. For example, next year at ASM Microbe, they'll be taking care of the recording and the cameras and the lights and all of that. So we appreciate them a lot. And they also let us be on microbeworld.org. I also want to thank the American Society for Virology. Every year at their annual meeting, they let TWiV do a live episode. And we greatly appreciate that, because we have a lot of fans there. And all the universities who invited us to go and hosted us during 2015, we really appreciate it. And I think back at one of these entries, I think it was Brian, one of his reasons for listening to TWiV, keep my dream of a live TWiV in Memphis replicating. Well, Brian, just tell Stacy to invite us. It's very easy. That's the rubric for getting people to do TWiV. You say, we invite you to do a seminar, and just have them do a TWiV instead. It's not very hard. And I want to thank all of you guys, Alan and Kathy, Rich, Dixon, and all the hosts on all my other shows for doing this. And especially this week, Alan and Kathy, it's New Year's Eve, and last week as well. It shows dedication. I couldn't do this without them. So thank you guys very much.
Well, and thank you for doing this, for getting this thing going, and the incredible work you put into editing it too.
I'm having a blast, I got to say. I have so much fun, and it's learning. And boy, I can just do this the rest of my life, and I will.
Great.
So don't worry, even if all my co-hosts leave, I won't do it myself. I'll just find new co-hosts, but I hope they stay with me for the duration.
I'm sticking around.
All right, very good.
Me too.
I think Rich is with us, and I think Dixon is as well. I don't know about the other shows, but they seem to like it, so I'm really lucky. We have great hosts on all these shows, and even the new TWiVo, Nels, is wonderful. He's just wonderful to co-host with. So
all a virus really needs is a good host.
Ooh, yeah, excellent. That's a good title. Okay, so now we will tell you 10 stories of 2015 in no particular order that we thought was cool. Each of us picked a couple, and we all will chime in and tell you about what we think. And one of the ones that I really liked this year was a story arc that really spanned multiple episodes, and that is recoding viral genomes, which was initially done to make vaccines. And eventually we did a paper where that gives you some insights into viral functions. In the first episode this year I'm thinking about is TWiV 351, which was called the dengue code. And there we talked about a paper where the virus was attenuated by recoding the genome. And this is not the first time this has been done. It's been done previously with influenza virus and polio virus. But this introduced us to this really interesting concept, which is apparently applicable to multiple vaccines. And I guess in the future we'll have at least one vaccine being licensed at some point that uses this approach, because you can have an infectious vaccine with so many mutations that it's not likely for them to revert. But then we got involved in the discussion about how this is working, right? What is the mechanism? Is it codon bias? Is it codon pair bias? Is it dinucleotide bias? And I think for a couple of episodes we had emails about that.
And it turns out to be mechanistically a really juicy question.
It's very interesting. Now, I just saw this week a paper by someone who has actually written to us. The first author is Dusan Kunek and Nikolaus Ostrider. Codon pair bias is a direct consequence of dinucleotide bias. This is in Cell Reports. Is this open access, by the way?
It is, yes.
All right. So this basically is a bioinformatic. No, I shouldn't use that word. Eugene Koonin will get mad at me. Computational biological analysis. Thank you, Rich. Of a variety of different viruses, their genomes. And their assumption is that human viruses that infect human cells, they have to use the codon utilization of the host. And so they analyzed the codon utilization of a variety of viruses, and they conclude that its codon pair bias does not play a major role in determining, but it's rather dinucleotide bias. So maybe we'll do this at some point in the future. Right. But I have a feeling the argument is not over. Nope. But this is certainly the latest contribution. Yeah, Dunek wrote to us in September, and we read that on TWiV. And then there was our paper in TWiV 362, where it is basically an analysis of herpes simplex virus gene regulation, which can be controlled by codon usage. And that was from... I'm forgetting his laboratory. Let me just click on the link here, because we have links in all the show notes. It's wonderful. Yes, Ron Desrosiers, Importance of Codon Usage for the Temporal Regulation of Viral Gene Expression. So I think this is the tip of the iceberg, so to speak. Now that people know this, they're going to look at it in all sorts of contexts, and this is going to be big. Mm-hmm. All right, that was one. My next one, we did a bunch of papers on defining the human virome. And that's all started with TWiV 323, a skid loader full of viromes. We have a nice illustration of a skid loader there full of virus particles. Changes in the human fecal virome associated with Crohn's disease and ulcerative colitis. Now, of course, everyone is interested in how the microbiome is associated with a variety of diseases. Just about any disease in the world, people want to associate with the microbiome. But I think the virome is equally important, right? Not only because it can regulate the microbiome, but there can be eukaryotic viruses in there that might influence it. So this study is an association, and we still need to find out causation, which we don't have yet. And, lots of studies on the microbiome are faulty in the same way, because they make conclusions that aren't yet supported. But I think virome studies are on their way now. We had a bunch of podcasts where we talked about that. 342, Public Epitope number 1. This was a cool discussion of a paper describing a method called VirScan, where it basically looks for all the antibodies you have to tell what infections you've had in your lifetime. So another way of getting at how many virus infections you have. And the cool thing there is that it's not perfect, is that they miss a lot of infections, and a lot of vaccinations that people have got apparently don't show up in the VirScan. So that's going to require more work. Then we had later on TWiV 355, Baby's First Virome, which I think was one of your former students, Kathy. Is that right?
Yeah, Ephraim Lim was an undergraduate in my lab.
Yeah. Yep. The fecal virome and microbiome in twins during early infancy. And the cool thing there is how it seems likely that the babies inherit their virome from amniotic fluid, and then that modifies the microbiome early on. So these kinds of studies are going to be more and more and more. Through TWiV 356, got viruses. This was the gut virome of children with serious malnutrition. I think we followed, that was right after the other one or shortly afterwards. Again, what can we learn in cases of serious illness? And 362, this is the one where I believe it's called Gotta Catch 'Em All. Yes, we talked about Lipkin's group assay to identify all the viral nucleic acids in a sample. That was pretty cool. So that's looking at nucleic acids as opposed to the VirScan, which was looking at antibodies. And so this one revealed quite a bit. And then finally, TWiV 365 was the last in this series, blood feuds and a foodborne disease. This was Thanksgiving episode appropriately. A new virus discovered in human blood by this, by sequencing human blood, basically. Right. And finding not only a human virus, but many other viruses as well. So we did a lot on the virome.
Yep.
And that's one of the things that stuck in my mind as I looked over all the episodes. I think this is in its infancy, and we're going to see more of it.
Definitely.
In the future.
This brings up a question, because I think I hadn't looked at the skid loader full of viromes episode image before, and you put those together. You don't outsource those, right?
Yeah, I put it together, yeah.
That one and the one with the mirror and all the yellow viruses on it, the rearview mirror. I especially like those.
Yeah, sometimes I do make one as opposed to, just finding one that fits.
Yeah.
Because I have lots of virus particles and I can just copy paste them. Although I try to use different ones so it's not obvious. Mm-hmm. Thank you. I do that, yes. All right, and the last one I picked out, we each picked 2 or 3, I think. I cannot let the year pass without mentioning, of course, the Ebola outbreak, which waned in the past year. Last year, 2014, we had lots and lots of episodes on Ebola because that's, of course, when it was at its peak. And I'm going to the Ebola sit rep for
me too.
December. So the total cases since the beginning, 28,637, 11,315 deaths. As of December 30th, no confirmed cases of Ebola reported in the previous week. On December 29th, WHO declared that human-to-human transmission has ended in Guinea.
Yes.
After 42 days with 0 cases.
But they will still be monitoring quite intensively for at least the next 3 months, is what I think I heard.
Right. So we talked about some of the aftermath. We talked about the vaccine that had been tested by the ring approach, TWiV 349, 1 ring to vaccinate them all. I think this is a really neat approach where instead of giving a placebo and the vaccine, you simply divide. So that the approach they took was to identify a case, and then all the contacts of that case, healthcare and family and casual contacts in the immediate area, were divided into 2 groups. One got the vaccine, the other got it 3 weeks later.
Right.
So it's kind of a neat way to not leave some people out and then have them get very sick and die. And we talked about this, and it looked good, but the numbers were small.
Right.
And then, of course, the outbreak basically dropped precipitously, so it wasn't possible to generate much data anymore. But I would guess that if there is an outbreak, and there will be at some point in the future, that they would use this in it to not only get data, but to contain the outbreak.
It's a nice approach. That's
a vesicular stomatitis virus vectored vaccine where the glycoprotein is replaced with the glycoprotein of Ebola virus. It's quite good. We also talked about some persistence of Ebola virus in the eye of a patient. That was TWiV 336, brought to you by the letters H, N, P, and I, EYE. And I have received an email from Lisa, who is a listener and who was my host at Berkeley a couple of years ago, and she heard this physician who had the eye infection speak, and she went up to him and she said, "There's this great podcast, TWiV, would you like to be on it?" And he said, "Sure." So we're going to get him on next year to talk about his eye. And then there was we also talked about persistence in semen and the possibilities of sexual transmission that spanned several episodes as well. And then we had 1 Ebola virus experiences, TWiV 341, where I was in Glasgow and I spoke with 3 individuals who were involved in some way. Emma Thompson, who had treated a nurse in Glasgow who had come back, and that nurse then had a recrudescence later on. And then Adam Kucharski, who had done some epidemiology, and Jillian Slack, who had gone to work in an Ebola treatment center. So if you want to hear people talking about how it was there. So that's Ebola for 2015. I'm sure we'll see it again. Mm-hmm. I don't know when, though.
Yep. Hopefully not in that scale again.
I hope not. That was a big one. I hope we're more prepared, right?
Yes, and it was a big one because it was badly responded to, and hopefully, the world has learned.
I also think that, what do I think? I've forgotten what I think.
I do that all the time.
Ebola will come back. It's just a matter of when. Oh, so in the area where it was just in Western Africa, I suspect that because we have these treatment centers, they can do diagnostics. They will be able to deliver vaccines effectively. I think it will not be as big as the previous one.
Right.
And I suspect that if it happens somewhere else, they will also go in with some of these vaccines. This is really what I wanted to say. As a consequence, we have a number of vaccines, not just this one that was that we talked about, but there are others in development. We have antivirals in development. We have monoclonal antibodies in development. These are going to be hard, of course, to test because there's no outbreak, but I suspect as soon as there is one, we'll go in there with a variety of these and get some more data and hopefully contain it.
Yep.
Okay, who's spillovers and scares?
So I did the next three. We had a kind of a story arc talking about various spillovers and scares, events where viruses either moved from animals into humans or some people thought they might. So, for example, the TWiV 347 Rose Rosette and Squirrel Roulette, we had this rather bizarre case of a couple of cases of fatal human encephalitis that were in squirrel
handlers.
People who bred, which introduced me to a whole new world. Wow, I didn't know people bred squirrels as a hobby.
Specific.
I'm not one to criticize weird hobbies, but
and specific kinds of squirrels.
Specific kinds of squirrels, and these were imported from the tropics to northern Europe, I think, and the variegated squirrel.
Yeah.
And apparently, as far as we can tell, may have transmitted a virus to some of their handlers, and a couple of these folks got very, very sick and died. And a very odd case. Doesn't seem to have gone human to human, but just an example of the kinds of things that you can stumble across when you go playing with wild animals, I guess.
Which we used to do a lot more in human history, but
yes, of course, because and I think we talked on the show about the first people to domesticate cats or pigs or what have you probably discovered some interesting zoonotic diseases. Of course, they didn't know what was going on, but that is one way to get contact with these. And then in TWiV 351, we talked about another odd case, novel pox virus that was isolated in an immunosuppressed patient. And I think nobody's quite sure where that came from, but there it was. And a lot of this, too, is these are the types of cases that in the past the physicians would have shrugged and said, well, we don't know what killed your relative or what made you sick, and that's the end of that, and sometimes these things happen. But now we have the tools, and you can do these analyses, and we discover entirely new viruses.
I find these kind of really fun to do on TWiV.
Yes.
Right? They're rare, like the squirrel spillover is pretty rare, but still it illustrates principles, and it gets you primed. You learn the methodology, and then you're ready in case you see it again.
Right. Mm-hmm. And then there was TWiV 364, which I wasn't on, but you had a couple of guests who came in and talked about a SARS-like, emphasis on like, bat coronavirus. And the title of the episode, it's not SARS 2.0.
Yeah, it was a good episode. I like that.
Yeah, that was a good episode. And this was a subject that had been covered in some rather unfortunate ways in the general media, and you can trace a good bit of that coverage back to an unfortunate press release from UNC Chapel Hill.
Yep.
That implied that this was a virus that was either jumping to humans or was about to, and was had the potential to be the next SARS, and really drumming up the hype without a whole lot of basis. So I thought that was a good corrective episode too. And then I highlighted as well TWiV 365, blood feuds and a foodborne disease. Just another case where you've got a virus of unknown origin that shows up, and again, because of these improved techniques, you can now see stuff that we wouldn't have seen before. So that was that arc. And then I couldn't let the gain of function debate pass, because that is something that came up pretty frequently.
Absolutely, and it will continue, and it
will continue to come up. So one of the things that I've linked to here is a Science Magazine news story that was stimulated by the recent release of the NSABB's preliminary report on this topic. because the report that they commissioned, the study that they commissioned to analyze the risks and benefits of gain-of-function research, as it's sometimes been called, finally came out. And that document by Griffin, consulting company called Griffin, is also available online and is linked to, I believe is linked to from the Science article.
Yeah, it is.
That is a very, very extensive, it's about 1,000-page document. And it's available freely online, so, go for it, dive into it. I read the executive summary and partway into it, and it actually seems like a pretty balanced analysis, and they put a heck of a lot of work into it. But this is a subject that we talked about several times. So I've got TWiV 321, where we had Paul Dupré on. He was talking about the moratorium and the terminology behind it. He favored some terminology that I don't think gained much currency, which was this A-trip.
Nope, never went anywhere. Even though we put it in our title, I figured,
Yep. So A-trip and a pause, and nope, it's just called gain of function.
Yeah.
But it was worth a try, and it was a good discussion, I thought.
Mm-hmm.
And the dengue code, TWiV 351, we talked about H1N1 and the gain of function debate.
Mm-hmm.
And TWiV 354, the cat and the heart. And then I forget whether there's a specific thing that stimulated that or if we just were riffing on.
Yeah, why gain of function experiments are valuable.
Right.
I think, it was based on an article in Science.
Okay.
Why we need flu gain of function research.
Right.
I don't pass up an opportunity to
no, clearly
talk about why we need this stuff.
And then TWiV 363, Eat Flu and Dyad.
Hey, we have good titles,
We do sometimes, yes. The focus of that, the title focus of that was this ferret study. There we go with the ferrets again.
Mm-hmm.
Spreading, how the mammary gland might have fostered spreading to nursing pups of the ferrets. So that selected for more transmissible flu viruses. So here we're selecting for more transmissible flu viruses in ferrets, and oh my God, the sky is going to fall.
Yep.
But a very, very interesting paper with some important implications, I think.
Those are both very nice papers. Yeah. I think in some future episode we should at least summarize this report from the consulting company.
Yes.
About gain of function. It is actually more balanced than I had expected. And
yes.
You can tell that by reading the summary, but you can also tell because Mark Lipsitch doesn't like it.
Yes.
He says it's too restrictive.
Yes.
Not enough experiments would be restricted if we listened to them. And now I want to pick on Mark even more than I have. He says in this Science article, that I am pleased to see an acknowledgement that some experiments should not be done, but fear that the exaggerated perception of benefits would neutralize the acknowledgement into a de facto anything goes approach. So here's the problem with Mark Lipsitch. There is no exaggerated perception of benefits. He and others said the SARS work by Ralph Barrick, which we covered on the episode, It's Not SARS 2.0, he and others said this work has no value at all, when in fact it had huge value, as we talked about on the episode. So, and I have pointed out before that Lipsitch is completely misunderstanding the value of some of these experiments, yet this quote makes it into the article, an exaggerated perception of benefits. And I think we have to hammer on this in the next year, that it's not an exaggerated perception, that and in fact you can't often predict what's going to happen, but certainly when you have the results, you can say, look, this is what we have learned, but he doesn't believe it.
Yeah, these are real benefits. They're not
right.
Exaggerated perceptions.
Right, and I think the problem that Lipsitch is, he's gotten himself into a situation where he was vehemently and vocally and publicly opposed to a set of experiments that are now widely acknowledged by the scientific community to have been A, not all that risky, B, conducted safely, and C, pretty darn useful.
Yeah.
So the original H1N1 work that stimulated this whole brouhaha, he was big into that debate, saying, "No, this never should have been done. We can't do anything like this." And now, if he were to acknowledge that there are reasonable benefits to some of these studies, he would have to go back and say, "You know what? I was wrong about the H1N1 stuff." And apparently, he's just not willing to do that.
No, not at
all.
So
I think that's the situation he's gotten himself into, is that he has convinced himself that that particular study could not possibly have been acceptable, and therefore nothing of that ilk could possibly be acceptable, and there's no resolving that. I don't think he's a reasonable participant in the discussion. But I was very, very impressed with the Griffin report. I liked the fact that they really went into it, and yes, absolutely, there are certain types of studies that shouldn't be done. I would agree with that. I don't think that aggressive bioweapons research is a good idea. I don't think it's nearly as dangerous as a lot of people would have you believe, but I think there are some things that just ethically we shouldn't be doing. And at the same time, it delves into the benefits of these studies, and I think it takes a very realistic approach to the risks.
I want to just quote one sentence from this Science article. After examining a number of scenarios, the report concluded that, quote, only a small fraction of laboratory accidents would result in a loss of containment. Of those, only a small fraction would result in a laboratory-acquired infection, and of those, only a fraction would spread throughout the surrounding community. And this is exactly what Lipschitz does not want to hear.
Right.
All right. So we will cover this in more detail in the future. This is going to be an issue all throughout 2016. It's going to be an issue in 2016,
for sure. Yes.
Because I believe there's another report of another. They have two consulting firm reports on the way out. This one and then a second one.
Right.
And then sometimes next year, I believe by April or so, the final decision will be made. And Ken Burns has been involved in this, and he said at that point he would come on and talk about it with us.
That'd be great.
He has been on the NSABB. I don't know if he's still on it as well.
Right.
Okay.
Right. And so number 6 now is the third one that I picked, which was just a couple of episodes really, but then also kind of a general arc that we've had, I think. So I wanted to highlight a couple of episodes where we really delved into detailed mechanisms of pathogenesis, and TWiV 336 brought to you by the letters H, N, P, and I. One of the other, so there was the Ebola virus story, but then the main paper that we had there, was it a snippet or the main paper? Anyway, it was a paper that had an influence on me. Was the
sometimes it doesn't matter anymore.
It doesn't matter. Yes, we could spend half the episode talking about a snippet. The interferon pathway mutations that led to severe influenza in a child.
Ah, yes, right.
And that patient was designated P. And I thought it was just fascinating that we now have the ability to drill down into a case like this and not only say the cause of this was the influenza, but to say the cause of this was the way the virus interacted with the specific set of mutations in the interferon pathway in this child. And that's hugely useful for understanding the pathogenesis of the virus in people who don't have those mutations. Mm-hmm. And I really liked that. And then the other one was TWiV 345, how a vaccine got the nod. And this was where we talked about the Pandemrix vaccine, which was linked to an elevated incidence of narcolepsy in some populations where this particular flu vaccine was distributed. And the manufacturer went and investigated, well, first of all, did our vaccine cause this? And secondly, all right, it looks like it is causally linked. Now let's figure out what the heck went on. And they went and took apart the whole mechanism that appears to have led to an autoimmune reaction in a subset of patients, and they really dissected what was causing that. And that is hugely informative for vaccine development and also for immunology, I think.
Yep, for sure.
I thought it was a really interesting analysis and a really good example how these sorts of things should be investigated.
I think it's important to point out also, this is not fodder for the anti-vax crowd. No. This shows you that when a rare adverse event happens, it gets sorted out.
Yeah.
And in this case, it's published. There's no denying it. And whereas all the other stuff that people are saying these are horrible adverse effects are not. They're just things that happen coincidentally.
Right. And when a real adverse effect happens, it gets investigated to pieces, as this is an example of. And it's like aviation. It's the safest form of travel ever devised is traveling on the airlines, and every now and then one of them crashes. And that does not prove that the airlines are death machines, because what you look at is after that happens, there's this incredibly detailed investigation that grinds through every little bit of it and says, aha, we figured out this was the cause, and then implements procedures and such to address that particular cause, and that's how you get something that's that safe. And that's what's done with vaccines. You've got a whole system for monitoring it, and when something goes wrong, it's examined minutely, and it gets us to this type of understanding. So those two episodes were ones where we talked about this type of analysis, but then we also had a whole bunch of episodes, 324, 328, 329, 330, 339, 343, 349, 353, 369. Those were just the ones I picked out going through for the year. And those were all episodes where we talked about how basic research, a lot of the basic research that goes into these types of discoveries. So the reason we're able to figure out that P's influenza developed in this particular way and that this particular vaccine caused this is because for decades people have been doing the kinds of basic research that we talked about in these other episodes.
Yeah, it's important to point out how important basic research is.
Right.
Every clinical advance has its roots in basic research, whether it's a vaccine or antiviral or an assay that you use to measure infection, to do epidemiology. You need to have the basic research first.
Yep.
And we're just not ringing our own bells here. This is important. It's important stuff. And I think the people who do the clinical research realize this as well. Yeah, I think the basic stuff is wonderful, although we do try and mix it up on TWiV, right?
Absolutely.
To show you the whole entirety of virology, although I know we're not perfect. We miss out on a lot of viruses. We don't do a lot of phage, although we did one recently. We don't do a lot of plant viruses, although we did do one this year.
Yeah.
And we listen, if you tell us do more of this, we'll try and do it. All right, Kathy, you have a couple.
Okay, yeah, I didn't choose themes or arcs with multiple episodes. And by the time I got to the list, there were only 4 slots left, so I took
Sorry
2 and left 2 for Rich. But the first one was I think the very first one where we had a new episode in last year was TWiV 319, where we talked about the results of a large tetravalent dengue vaccine trial, and that's this vaccine called CYD-TDV. It's a live vaccine based on the yellow fever virus backbone, and it expresses 2 viral proteins, PRM and E. He swaps those genes out from the yellow fever virus and puts in all 4 serotypes from dengue. And the efficacy of this for patients just getting 1 injection was highest for types 3 and 4 of dengue, from 74 to 78%, and lowest for types 1 and 2, 50% and 42%, respectively. Nonetheless, this is thought to be a pretty successful trial. And the concern, as always with dengue, is that a person who's had an infection by one serotype, if infected by another serotype, can develop a disease that's very severe because of the way that there's this antibody-dependent enhancement of infection. And so the concern is if you don't get complete protection, for instance, for type 2, then will this vaccine be bad for an individual if they get type 2 after getting this vaccine? And this vaccine is now called Dengvaxia, and just in the month of December has now been licensed by 3 countries, Mexico on the 9th, Philippines on the 22nd, and Brazil on the 28th.
Awesome.
Yeah. So that was one paper. And then the other paper that we were going to discuss on that episode, and we never got to do.
It's been at the bottom of the show notes ever since, all year.
I kept saying, are we going to do that other dengue paper? But I'll just summarize it for you. It was pretty cool. It's a group from, I think, 3 laboratories, Felix Ray, Euthytep Microcephaly, and Gavin Screeton. The last, those last 2 groups are from Imperial College London, and Felix Ray is Institut Pasteur and CNRS. What they did was look at antibodies from patients that were highly viremic with infection of dengue virus. So they had lots of dengue in their blood, and they isolated antibodies from those patients and then characterized them and found broadly neutralizing antibodies that could neutralize all 4 serotypes. And they were targeted at the envelope dimer epitope that bridges between 2 protein subunits of the mature virion. So this is really cool because it implies that if you could develop these kinds of a vaccine that would elicit these kinds of antibodies, then you would get that broad protection against all 4 serotypes. And that group then went on and in May published a paper where they looked at the X-ray crystal structure. The first author of that paper is Ruvinsky, and the same last 3 authors. And I forgot to say the first author of the paper that we would have covered in January was Dejner Radisai. Anyway, they have the X-ray crystal structure and show that E-dimer epitope interacting with antibodies. And they have antibodies from, they do crystal structures of, I think, 4 different patients. So it's a pretty cool story because it's leading to additional dengue vaccines. And
Yes.
In addition to the one that's just been licensed in those 3 countries, there are other dengue vaccines that are either in phase 1 or phase 2 trial that are recombinant chimeric. And Merck has a phase 1 subunit that's expressed in Drosophila. Something based on these papers I just described would also be considered a subunit vaccine if you just gave the dimer interface as the epitope or antigen. So I think there's going to be even more progress in dengue vaccines, and that bodes well for other things, perhaps like Zika, another flavivirus, and things like chikungunya as well.
And by the way, since we waited so long for that first paper, to cover that first paper, it was published in Nature Immunology. I think it came out in print in the February issue. And it has now become public access because it's been, I guess, more than 6 months or whatever the rule is on that. So you can go to PubMed Central and read it on there.
Oh, good.
The second paper is not open access yet.
Right. So there's some virtue in our delay.
Yes.
We used to have all these papers at the end of the show notes, and I took them out. I have to put them back because we actually had a letter from someone asking some good questions about this dengue trial that we had covered that and asked us to go back over it. And I really want to do that at some point in the future, so I'm going to make a note to remember that.
Okay, and then the other thing I picked was kind of along the lines of what Alan was saying, where papers looked at very basic questions that kind of have been bugging people for a long time. So I picked the paper we covered in TWiV 340, which was called No Shift Measles. And that was the paper by Fulton and several other authors, and the senior author was Nicholas Heaton. And it was the one that had the episode image of the airplanes in World War II, and where the bullets had hit the wings was where you didn't need to put armor.
Yes.
And this was the same kind of idea, asking the question, why do some RNA viruses whose polymerases have a high error rate have not very much antigenic variation, and you can have the same measles vaccine for years and years and years, versus other RNA viruses like influenza have lots of antigenic variation, and you have to make new vaccines every year if you want to try and target those that are circulating, those viruses that are circulating. And so what these authors did was introduce mutations into the measles genome and then look to see which of the mutations they could recover. And there were certain regions of the genome that could tolerate mutations, and there were certain regions that don't tolerate mutations in measles. And the ones that don't tolerate mutations would be the ones that you can't rescue those viruses. And I'm trying to make the analogy back to the airplanes and the wings, but not able to do that at the moment. But anyway, so it addresses that question, but as we kind of came down to at the end, it doesn't really answer the question for us. It to again come back to another meme. It just takes us one turtle down.
Yes.
So we now know that there are these certain regions in the measles virus that just can't tolerate mutations. The polymerase, for example, but that there's this flexibility or tolerability for mutations in some of the other regions.
Right. So there are some places you can shoot and it still makes it back home, and other places you shoot them and it doesn't make it back home.
Right. Right. And so along those lines of basic questions that answer long-standing basic approaches that or try to get at answers to long-standing questions, I also wanted to highlight the one that Alan just mentioned, 369, and it's how a neuronal pathway reactivates in herpes simplex virus. And again, I think that's where we came up with the turtles all the way down reference. Right. But so we think that there's this neuronal pathway that's involved in reactivation, but then what stimulates that neuronal pathway is the next question. Another one was, again, on herpes virus, a basic question: how does herpes simplex virus cause host cell shutoff? And there was some answer to that. It does it to some extent by disrupting transcriptional termination. Mm-hmm. So there's still these questions that students ask us, or we have to say in lectures. We still don't know the answer to this, but people are chipping away at them with some interesting approaches.
Okay. Cool. Yeah, we used to just pick individual TWiVs for these, but then at some point we got thematic and doing arcs. Either one is fine. It's absolutely cool. Rich sent me a list of his 10 top. I think he sent it to everyone. Mm-hmm. So I picked his first two and put them in here. And he picked 2 episodes, TWiV 346, which was recorded at ASV, A Double Helical Career, which of course was our conversation with Joan Stites. Great episode. Really, really good. At one of the best for sure, because she has had such an amazing career, and she's really, really good at talking about it and answering questions in an engaging way. And you ask her a question and she goes, "Okay." She knows exactly what you want to hear. And of course, she was Rich's PhD advisor. Yes, PhD advisor. He was her first student. Is that correct? I don't remember.
I think that's the case. So, he's shouting at the podcast now.
Yes, he's shouting at us. I told you that.
Yeah, well, we don't remember things. Anyway, I can understand for both reasons why you'd pick that as your number one. That was really great to do that. And he also picked TWiV 345, How a Vaccine Got the Nod, which showed up on Alan's list as well, for this story about narcolepsy caused by pandemics. Yeah, I think he really liked that as well. And that's 10 top things from 2015. One thing I noticed that didn't make our lists, but we talked about a lot, publishing, science publishing.
Yes.
And I suspect we'll be talking about that for years to come.
That's going to be coming up a lot.
Because it's got problems and it's not fixable really quickly.
Nope.
I want to just read one email because we're running late here. Not late, we're running a long period, and we don't want to go too long. But it's a Zika email, and I think it goes well with the previous one. This is from Helen, who was written to us before. She's an assistant professor at the University of North Carolina, Chapel Hill. Thanks for your recent discussion about Zika virus. The emergence of Zika in South America may provide an interesting example of how a previously described pathogen can have unexpected effects when it is introduced into new populations and new ecosystems. I'm particularly interested in the consequences of Zika infection in people who have previously been infected with dengue. Dengue virus is highly prevalent in Brazil. Most people are seropositive for one or more types. As you mentioned in your discussion, the antigenic cross-reactivity between Zika and dengue viruses complicates diagnosis. This has major implications for large-scale dengue vaccine trials ongoing in the region, so there is an urgent need for reagents that can serologically distinguish Zika and dengue. And I would add that now that the vaccine has been licensed in Brazil,
yes.
Is that correct? Is that one of the countries?
I think that was one of the countries.
Yes.
Yeah.
September 28th.
It'd be more of an issue, right? Wow.
Right.
The massive increase in microcephaly cases that has coincided with the emergence of Zika in Brazil is alarming, and it is important to determine the cause. It would be very surprising if Zika virus turns out to cause microcephaly, although a number of different viruses can cause congenital abnormalities, including microcephaly. This is not something that has been associated with flaviviruses previously, and if dengue virus produced microcephaly at any reasonable rate, we would know about it due to the large number of infections worldwide. So, if a causal role for Zika virus and microcephaly is demonstrated, this would represent a new presentation for Zika virus and flaviviruses generally. One question is why we would observe microcephaly in this current outbreak of Zika infection, but not in previous ones. One thing that is different about the current outbreak is that Zika virus infection is occurring in an area of dengue virus hyperendemicity, i.e. multiple serotypes of dengue co-circulate, so Zika virus is infecting people who are already seropositive for one or more or multiple types of dengue virus. The antigenic cross-reactivity between Zika and dengue may create the potential for antibody-dependent enhancement and new clinical presentations, as is the case for infection by different dengue serotypes. We have studies planned to address all of these questions, so stay tuned. I should note that I think in our previous letter, he mentioned that in the Polynesian, they had gone back and looked at the records and saw some... Here, French Polynesia, they went back to the records and noticed an increase in microcephaly in 2013 and 14. Right. So it may be that it has happened previously, Helen, but we just didn't recognize it.
Yes.
Well, we will look forward to your findings, and
yes,
we would love to talk about them.
And Helen, I spoke to a couple of months ago for an article I did for Nature Medicine.
Cool.
On chikungunya.
Chikungunya.
She is an assistant professor of microbiology and immunology at UNC Chapel Hill.
Yep. All right, let's do some picks of the week. But before that, let me also tell you that this episode of TWiV is also sponsored by the ASM Biodefense and Emerging Diseases Research Meeting, which is taking place February 8th through 10th in Arlington, Virginia. This is a meeting sponsored by ASM, and you can register for early bird rates up until January 14th. So just like ASM Microbe, where you have to get your abstract in soon, here, to save some money, you should register by January 14th. This is the premier event to discuss how to defend against bioterrorism and shape the future biodefense research agenda. It's also got a large component of emerging disease research as well. You can learn more about this meeting at asm.org/biodefense2016. Alan, what have you got for us this week?
I have a store I used to shop from all the time when I was a kid. And I just recently discovered that they are still a going concern. I used to spend hours staring at the Edmunds Scientific Catalog. I don't know if you remember this, but they sold.
I loved it.
It was awesome, and they were most known for their optics, which I think is where they got their start. But they had telescopes and microscopes and hot air balloons and all sorts of just really cool. This was back before shopping malls had the designated science store.
Sharper Image and
right, and most of those places don't even carry a lot of science related stuff. But Edmunds Scientific, you could get the really cool stuff. And a lot of my allowance and my Christmas wishes were centered around that catalog. Anyway, I recently had occasion, I got a hold of an old microscope, very, very old microscope. It was my grandfather-in-law's, and didn't have any slide making materials of any kind, and wanted to show my daughter how to use this thing. So I needed to find microscope slides and cover slips, which was never a problem when I worked in the lab. But now that I'm not in the lab, where do I find these things? And there are some sources, but they either, you either have to buy a case, or they're, not convenient, or they're run by nuts or what have you. And then I found Scientifics Online, which is apparently Edmund Scientific branched off. They have an optics company now that just does lenses and optics. And then Scientifics Online is what I used to associate with the Edmund Scientific catalog. It's all their other stuff. So this is up and running, and I ordered from them, and they are very prompt with the order processing, and I've got my slides and cover slips ready to go, and they have a lot more stuff too.
Oh, I remember that catalog. It was great.
Yeah.
And in fact, not too long ago, I used to get the optics catalog here.
Right.
With all the lenses and scopes and so forth. I bet you could get a prism that does birefringence there.
Probably
good. Yes.
One of the top featured products is a pendulum wave desktop model that I think would be good for Rich.
Yes.
Another place I used to love, Alan, was Heathkit.
Oh, yes.
I think you might have even picked that once.
They rebooted the company. Some people bought the old company and rebooted it recently, but it's a shell of its former self.
In fact, when I first went into Peter Palazi's lab as a PhD student, he needed power supply, so I ordered 2 Heathkits, and I put them together really quickly because I knew how to do that.
Yeah.
And I spent a couple of hours, and I gave them to him, and he said, "This is great, but you shouldn't waste your time doing this. Do experiments," which is the way Peter was. Kathy, what do you have?
I picked something that I found as an astronomy picture of the day, and it was a Vimeo staff pick. It was on December 22nd, and I put in the link to it. These people put a camera on the roof of the Exploratorium in San Francisco and recorded an image every 10 seconds from before sunrise to after sunset in mid-2009 to mid-2010. And this Vimeo captures 360 of those in a nice grid, and you can play it. I encourage you to put it on the full screen and move your cursor out of the way, because in the bottom right, then you can see the time of day. And then each one of these squares, you get to see whether there's clouds or there's water on the lens. Or if you watch carefully, you'll see a couple people in a couple images kind of in the lower right third, kind of peering around the camera just briefly. But you can clearly see how the long days and long nights change with the year and time of year and so forth. And it's just, I thought it was really cool for the end of the year to see almost really a year's worth of images.
It's very cool, yeah.
And the music is nice and, yeah, so.
All right, my pick is a giant squid in Tokyo Harbor. This has got to be the coolest video I've seen in a long time. Giant squids, as many of you know, get really big. They live way deep in the ocean, and up until a couple of years ago, no one had ever seen a living one. Am I correct to say that, Alan?
I think it was just a few years ago that finally an ROV crew or somebody had a deep sea video of one, yeah.
So a couple of days ago, one wandered into Tokyo Harbor, and they sent down scuba divers to help steer it out, and they took incredible videos of this thing. Look at the eye. Oh, my gosh, yes. This is so beautiful. And the tube below it where it pumps fluid. Oh, man, to go forward. This is just the most beautiful thing I've ever seen. It's only 12 feet long, apparently. It's this tiny one. But the tentacles, the coloration of the skin, it is just amazing. So I'm sure most people have seen this because it's been all over the news.
Right.
But I hadn't seen it till I saw it in the show notes, so I appreciated it.
This thing, it opens and closes to pump water out. Yeah. And it goes forward when it does that, right?
Right.
Oh my gosh. And the tentacles are just awesome. And this is life and its amazing beauty and complexity, man. And I just want to know what viruses are in this thing.
Yes.
Anyway, it's one of these things that just gives me chills because you don't see this very often. And it's pretty cool. We have some listener picks. One is from Daniel. Dear TWiV doctors, I saw this and thought of TWiM, but since TWiM doesn't do picks, I figured I sent it to you. And this is a link to Paper Life. The artist Rogan Brown cuts thousands of microorganisms in paper. Beautiful.
These are gorgeous.
Fabulous.
Beautiful. He cuts them.
Yeah.
Yeah.
So we'll take this anytime on TWiV. Is this worth a set of your books? Didn't think so. Anyway, I want to keep my amateur status so I can proselytize TWi with a clear, unbribed conscience. I just realized that I didn't sign it. I guess I've written in pics enough to assume familiarity, so let me sign it. Dan. P.S. I too loved the See Androids Fighting reference. I am a huge Rocky Horror Picture Show fan. Stephen writes, he sends a link to an Ars Science Q&A, how to deal with science denialists. And I think this is funny. He starts out by saying, the first question, it's a flow chart to help you deal with them, is whether you're dealing with a fully committed denialist. You're never going to get Peter Duesberg to admit HIV causes AIDS, etc. So it's another way of approaching this, which we've talked about many times on TWiV before. And then we have one from Venkat who writes, Dear TWiVistas, here's my listener's pick. It's a software called Cabisa, developed by the Institute of Tropical Medicine at Antwerp. A nice learning tool for clinicians involved in tropical medicine practice. It's quite detailed and not like one of those iTunes apps for whiling away your time. So it's a training tool for diagnostic skills. So you can figure out what someone has. It's an app you download.
Cool.
We also had a pick earlier in one of our emails, if I recall. It was from
Muhammad, I think.
Esper. Oh, was it Muhammad?
Oh, no. You're probably right.
Esper sent a link to, yes,
It was
an article in the New Yorker on the Fold Scope project. Thank you also. It's 4 listener picks today. Well, what the heck? It's the end of 2015.
Right. You guys got to pick a zillion things for your top 10, so.
Right.
We did.
Is there any episode that didn't get picked?
We might have picked every one of them, which is kind of... it's a year in review.
It's a year in review. Yeah.
Maybe I should call it a year in review anyway.
Instead of picking a few episodes, we could just say, let's review the year.
Yeah, that's true, because that's what we did.
Because I based my picks on yours, and you seem to be picking arcs rather than episodes, so I picked...
Whatever we want. It's all up to us because we make no rules. This is TWiV, which you can find at twiv.tv and very soon to be permanently at microbe.tv/TWiV sometime in the next few weeks. And we do love getting your questions and comments. You might as well start sending them to the new email, twiv@microbe.tv, although you could still send them to the old one. If you love it and you don't want to change, I know a lot of people love old things. Someone wrote me an email this week and said, I don't like your new icons. They're too cartoony. Okay, you're always going to not like something. I understand that, but change is good. So send your questions and comments to twiv@microbe.tv. Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun, both today and all year long.
And have a happy new year.
Thank you. You too.
Enjoy. Alan Dove is at turbidplaque.com. He is also on Twitter. Thank you, Alan.
It's been a pleasure all year.
And Happy New Year to you as well.
Yeah, Happy New Year.
And Happy New Year to Rich Condit, to Dixon Despommier, and to all our listeners. Be safe, and we will see you in the new year. I am Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.